So, welcome to the immunology lectures, and today we will start with one of the very important effector systems in the immune system. We call it the complement system. So, the complement system is one of the major effector systems of the humoral branch of immunity. By now, you have known what humoral immunity means, what is the how the innate system and the adaptive system they work together. So, the humoral immunity, which is primarily the B cell based immunity and which is mostly mediated by the antibodies. Now, what do these antibodies do? We told in our classes in humoral immunity that these antibodies can do many functions like neutralization, opsonization, but how were these functions mediated? So, one of the very important effector system of the humoral branch of immunity is the complement system. The complement system, although it looks may be a bit complicated, but actually I will try to explain it very clearly and lucidly to you, so that you get a idea how these complement proteins they act as part of this effector system, and they actually kill the pathogen, or they do many other functions. They can also elicit inflammatory responses. So, how was this complement system discovered, and what exactly the role of the complement system is? So, although the complement system, as I told, is the major effector system for the humoral branch of immunity, there can be complement systems or complement activation which does not depend on the antigen antibody reaction. So, a branch of complement activation starts from the interaction between the antigen and the antibody. So, when there is an antigen antibody interaction, there is complement activation, as well as there are antibody independent complement activation pathways. Before we come to these pathways or describe these pathways, we first try to understand or try to know what these complement proteins are and how they work and what they do actually. So, the discovery of the complement proteins, and as you can understand the name, that it complements something, that means it completes some incomplete job. Now, what does it complement for? So, back in like 1890s, there was a very interesting discovery from a very talented scientist, Jules Bordet. So, what he found was that he took the antiserum. You know what the antiserum means. So, the antiserum contains the antibodies. So, he took the antiserum from a Vibrio cholerae infected person. So, took the antiserum, and using that antiserum, he applied that antiserum on the bacterium. And when the antiserum was applied or given on the bacterium, he found that the bacteria was eliminated. That means it was dying. So, the bacteria was killed. So, now definitely the antiserum contains something which is able to kill that bacterium, and that something is probably the antibodies. So, of course, the antiserum means it contains the antibody to that pathogen, but now what he did was, so he heated that antiserum, he heated it, raised the temperature, tried to denature the protein components inside. So, if you heat it, the antiserum, there will be denaturation of the protein components. So, it was denatured and used that antiserum against the bacterium. He found, as expected, that the heated antiserum was clearly not able to clear that infection or kill the pathogen, kill the bacterium. Now, what he did was he took another fresh serum. He took some fresh serum, not the antiserum, remember. So, it was not the antiserum, the antiserum was heat inactivated. So, it was heated and supposed to be inactivated, and it does not work against the bacterium. Now, he took some fresh serum and added to that heat inactivated antiserum, and he found that the fresh serum is expected not to contain any antibodies, and because antibody is present only in the antiserum. So, the fresh serum that he took does not contain any antibodies. So, it is not expected to work on the pathogen. So, now he found that when he added this fresh serum, it again became competent to kill the bacterium. So, that was a very interesting finding. So, then there must be something in that serum which is complementing the function of the antibodies which are still present in the antiserum. So, heat inactivation actually killed or destroyed some components or some protein components which are not the antibodies. The antibodies could still survive the heat treatment, but those proteins were very labile to the heat treatment. So, they were destroyed, and upon addition of the fresh serum along with the antiserum, again regained the bactericidal or the bacteriolytic activity. So, the cells that mixture of that inactivated antiserum and the fresh serum could actually lyse the bacterial cells or kill the bacterial cells. So, this was an interesting finding. So, then he thought then there must be something in that fresh serum that is complementing for the inactive serum or the heat inactivated serum. So, the heat inactivated serum is supplying the antibodies which were not destroyed due to the heat treatment, but some other proteins which were serum components which are normally present in the serum, they were destroyed. Now, when you give this antisera, when you give this fresh serum, those components come in and they can now together with the antibodies from that heat inactivated antisera, they together can complement for this function and can lead to the lysis and killing of the bacterium. So, this experiment clearly established that the serum contains something which or some protein components or some components which are very much heat labile. So, they are susceptible to heat or sensitive to heat, but their presence is important for the antibodies to work against the pathogen. Later on, some years later, another scientist called Paul Ehrlich. So, Paul Ehrlich, he actually later on worked on the same components, and he coined the name for these components as the complement system or the complement proteins. Now, what does this complement protein means? That they can complement the function, they can complement the function of the antibodies and that completes the function of the humoral branch of the immunity. So, that is the function of the complements. So, these complement proteins later were studied very well, and of course, this person Euler Bordet, he received Nobel Prize in physiology and medicine later on, and this was very thoroughly studied later on, and people found that there are at least 30 different types of complement proteins that are present in our system, and they are circulating proteins. So, they are usually proteins or glycoproteins, and they circulate in our system all the time in the blood. So, they are always present in the blood, but they do not act. They only act when there is a pathogen present. So, they will act only in presence of a pathogen or in presence of an antigen antibody interaction. When there is an antigen antibody interaction, they can start acting. So, what do these complement proteins do and what they are actually? Before we go into the complement activation pathways, so these complement proteins, as I told, these complement proteins, there are 30 different proteins and they move around in the they moves around in the blood. So, let us say, so this is the in the bloodstream. So, there are these complement proteins which move around in the blood, but they are in inactive form. So, they are in an inactive form. So, they are not active. What happens is, whenever there is activation of the complement system, whenever there is activation, these proteins, they start activating each other, and this activation is primarily based on protease activities. So, these are somehow these proteins, they have proteolytic activities or protease activities. So, they can cleave each other, and these proteins, these complement proteins, they are cleaved and they get cleaved into a small subunit and a large unit. So, now these small and the large fractions, that are the cleaved fractions, they are actually the active complement proteins, and now they start to associate with other complement proteins, right. So, once they are cleaved, they get activated, they find another target protein and then associates with that target protein and starts to activate another complement protein. So, this is a cascade. The activation of the complement system is a cascade of events. It goes on as a cascade, as a chain reaction, one after the other, and that activation leads to finally a certain killing of the bacterium. So, now these proteins, as I told, they are moving around in the blood all the time in inactive form, and then they are activated by the cleavage. Now, where do these proteins come from? All these proteins, they usually come from the major source of this protein is the liver, and also to some extent from the tissue macrophages, and to some extent from the epithelial cells of the GI tract, and these are minimum sources. So, the maximum source, that is where it comes from, is the liver. Liver is the main source of this complement proteins. So, they come here and they go into the blood, they circulate in the blood, and whenever there is a signal primarily from antigen antibody interactions, they get activated, and once they are activated, they start cleaving each other. So, that is a very interesting phenomenon, that they start to cleave each other, and once they start to cleave each other, they generate active proteins or active proteases. Now, these proteases in turn cleaves other complement proteins, and this cascade of event keeps going on until there is killing of the bacterium, and how it kills the bacterium we will come to that, or how it kills the pathogen we will come to that. So, these complement proteins, just to understand the nomenclature, these complement proteins are named like, starting from, they are called like, named like C1, C2, C3, C4 likewise, and they are cleaved products. So, when they are cleaved, like for example, when C2 is cleaved, it can give rise to C2A and C2B. Similarly, when C3 is cleaved, it can give rise to C3A and C3B. So, usually when they are cleaved, the smaller fragment is denoted by this term A. This is a smaller fragment. So, if we call this is a complement protein and it is cleaved like this. So, the smaller fragments are usually the they are designated by the A, and the bigger they are designated by the B, or the larger fragments they are designated by the B. So, that is an usual convention. So, similarly, they can be cleaved into different fragments, and these fragments they can associate with each other and they can form new enzyme complexes which can cleave further another target complement protein, and this cascade of event keeps going on. So, likewise these complement proteins, as I described, they are named like C for complement, and depending on their identification, that like the C1, C2, C3, C4, they have been named like this, and the cleavage products are named like C2A, 2B, 3A, 3B likewise. We will come across these cleavage products and what do they do, how do they work, we will come later on slowly. So, now what do these complement proteins do? So, what do the complement proteins do? They primarily do three types of functions. One, lysis of cells. So, what is lysis of cells? So, the complement proteins, when after this cascade of events, they can form a structure like this, a cylindrical structure like this, which is also known as the membrane attack complex or the MAC. And this membrane attack complex on a target cell, for example, let us say this is a target bacterium, and on the surface of this target cell or on the cell membrane, this MAC or the membrane attack complex is inserted like a cylinder. So, it has a cylindrical structure like this, and on the cell surface or on the cell membrane, they gets inserted they insert this cylindrical structure and leading to formation of hole or pore on the membrane. Now, if you suppose if you are forming a hole on the membrane, that means that would lead to an osmotic imbalance, and that would lead to inrushing fluids. So, fluids will immediately come in, and that would lead to swelling, and the cells will finally burst. So, once there is an osmotic imbalance, if you have a structure like this and that structure goes into the cell wall or the cell membrane and creates a hole on the cell, then there will be fluids coming in, there will be inrushing fluids. Very simple, you are just making a small hole on the cell, and this structure is formed at the end of the complement activation process. And this structure is also known as the membrane attack complex because it attacks the membrane, and by that how the name comes from, it is called the membrane attack complex, which goes into the cell membrane of the bacterium, leading to the damage and leading to inrushing fluids, and finally the cell lyses, it kills the cell. So, it lyses the cell and it kills the cell. A second way the complement protein act is known as opsonization. If you remember, we have used this term also in case of humoral immunity, that is antibody mediated immunity. So, opsonization basically requires the antibodies. So, it is a combined effect of the complement proteins and the antibodies. So, opsonization primarily, that means it helps or assists the phagocytes or the phagocytic cells like the macrophages. For example, another APCs. So, what happens is when there is antibodies, they bind to the target pathogen on the surface of the target pathogen, and as we also studied earlier, there are complement receptors. So, there are complement receptors on the cell surface, particularly this macrophage, this kind of cells, the phagocytic cells, they express complement receptors on their surface, and there are complement proteins as well. So, the complement proteins, they coat the pathogen and are recognized by the complement receptors. When the pathogen is coated by the complement protein, they are recognized by these complement receptors, as well as assisted by the presence of the antibodies. So, there are antibodies which coats or comes and interacts with the pathogen, and as well as it is complemented by the cleavage products of these complement proteins. So, for example, one of the very important complement cleavage products that mediates opsonization is C3b. So, they can coat the pathogen, and can initiate the process of opsonization along with the or in presence of the antibodies. And a third process that these antibodies that these complements can do is enhance inflammation. So, enhancing inflammation, and this is primarily done by the smaller fragments of the cleavage product, that is like the C3a, C3a, C5a. So, these are the C3a and C5a. These are the major mediators of enhancing inflammation. For example, we discussed in our classes, in the initial classes of inflammation, innate immunity and inflammation, we have discussed about the migration of the neutrophils, for example. So, the neutrophils, they can bind, they have on their surface, they express these receptors which can bind to this C3a or C5a, and they kind of act as chemoattractants, like we have described the chemokines as the chemoattractants. Similarly, these complement cleavage products, particularly the smaller cleavage products, they also act as chemoattractants, and they can also enhance inflammation, and also they can do a second function, that is the degranulation. So, they can also bind. So, these granulocytes, the mast cells, they can express on their surface these receptors which can bind to this cleavage product of the complement or the complement protein cleavage products like C3a, C5a, and they can lead to degranulation of the mast cells. Degranulation leads to the release of histamine. If you remember, we have told in one of our initial classes regarding the release of histamine, which actually leads to vasodilation and increasing vascular permeability. So, these are all assisting or enhancing the process of inflammation. So, what does this complement proteins they finally do after they have been cleaved? They finally do these main three things. One is lysis, cell lysis, second is opsonization, and third is enhance inflammation or inflammatory responses. So, these are the three major functions that the complement proteins can do. Now, let us come to the different complement activation pathways. Now, how these complements they get activated. So, we will be discussing about the complement. Now, we have to know the complement pathways. So, complement pathways means the pathways, that means how these complement proteins, they initiate the cascade of cleavage events. So, the complement pathways can primarily be of three types. One, the classical pathway. Secondly, an alternative pathway. And thirdly, also known as the lectin pathway. So, these are the three major pathways by which the complement activation occurs. The classical pathway, as the name suggests, it was one of the very first pathways that was being identified, and it is the major pathway or the major effector pathway for the humoral immunity. So, this is an antibody dependent pathway. So, it is an antibody dependent pathway. So, it depends on the antigen antibody interaction. It will not be initiated, the classical pathway will not be initiated if there is no antibody antigen interaction. The alternative pathway is an antibody independent pathway, and the lectin pathway is also an antibody independent pathway. So, this is an antibody dependent pathway, this is an antibody independent pathway, and this is also an antibody independent pathway. So, a classical complement pathway usually is activated only when there is an antigen to antibody interaction, and antigen to antibody interaction is initiated, then only it starts to work. Now, before we start understanding the different pathways of the complement activation, what are the components of these systems? So, for example, the classical pathway mainly is initiated by the C1, the complement protein C1, and the other components that are involved are C2, C3, C4, 5 likewise. So, now the main component is the C1, and this component C1 is responsible for binding to the antibodies. The alternative pathway does not require the C1 or the C2. The alternative pathway, as I told, is an antibody independent pathway. So, it does not depend on the antibody antigen interaction. So, it starts only when there is C3b present in the surrounding. It will start only in presence of C3b. So, that means C3 has to be broken into C3a and C3b. So, the alternative pathway can start in two ways. Either there is some spontaneous breakdown of the C3 into C3a and C3b. So, the C3 which breaks into the C3a and the 3b, and if there is this 3b available, then the alternative pathway will start. Alternatively, it can also start as a helper pathway for the classical activation. That means, when the classical pathway has already been initiated, then the alternative pathway can start as a signal amplification system. So, it can even amplify the whole thing, and the lectin pathway is primarily activated by certain lectins, that means which binds to the carbohydrates, and they are activated when these kind of lectins, like for example MBL or mannose binding lectin, they can they can bind to the carbohydrates, the mannose moieties, the carbohydrate moieties on the surface of the pathogen. So, they recognizes the carbohydrates on the surface of the pathogen by this kind of lectins like mannose binding lectin. There is also phycolin. So, these are the components of the lectin pathway, which is also an antigen or antibody independent pathway. It starts primarily by recognition of the carbohydrates which are present on the surface of the pathogen or the bacterium, and the components are pretty much similar to that what we see in case of the classical pathway. So, it also contains the C2, the C4, the C3 and the C5, and in this case, in case of the alternative pathway, we have two factors which are also actually complement protein. They are named like factors, factor B and factor D. So, these are the components of the alternative pathway. So, the classical pathway comprises of the complement protein C1, C2, C3, C4, C5 mostly. Then the alternative pathway comprises, it starts from the C3 only, the C3b, and it has the factor B and the factor D, and the lectin pathway, it is dependent on the binding of these lectins, the MBL or the phycolin, which can bind to the carbohydrates or the that are present on the surface of the pathogen, and they also have these complement proteins like C2, C3, C4, C5. Now, all these complement pathways we will see in our next lecture. We will see how all these complement pathways, they converge, and the main objective of this complement pathway, kind of the central part of the complement pathway, is the complement protein C3. All their efforts are towards breaking down the C3 into C3a and C3b, because you need C3b. One of the major components that you need for the opsonization process or the membrane attack complex formation, they are the C3b and the C5b. So, C3b is one of the central components. You need a lot of C3b. So, you need to break down the C3 into 3a and 3b, and that is the central part of the complement system. So, all these pathways, be it the classical or the alternative or the lectin pathway, they converge at a point and they all try to break down the C3 into 3a and 3b. That is the main objective, and for that these cleaved products of this complement system, they try to associate with each other and form a complex which also is described as a C3 convertase. So, their objective is to form a C3 convertase, and a C3 convertase basically converts C3 into 3a and 3b. So, we will see in our next lecture how these complement pathways, they try to produce this C3 convertase, and how this C3 convertase is actually breaking down C3 into C3a and C3b. So, this we will discuss more in details in our next lecture. Thank you.